Hi there, Dr. Sadaf Qureshi here, and today we are going to talk about mitosis. Let's start the topic with introduction. Mitosis is one of the process of cell division, the other is meiosis. It occurs as a part of cell cycle where cell prepares itself for division during interphase, then it divides itself during M phase. Let's understand the cell cycle in pattern. Cell cycle include interphase and M phase. Interphase is further divided into G1 phase, S phase and G2 phase. During interphase, cell prepares itself for division. Then occurs M phase, that is also called as mitotic phase. M phase is divided into nuclear division and cytokinesis. And nuclear division is further divided into 5 phases, that are prophase, prometaphase, metaphase, anaphase and telophase. As today we are going to talk about mitotic division or M phase, so we will talk about each of these steps one by one. The first step that happens during mitosis is prophase. Chromatin is an uncoiled form in the start of prophase of M phase. Then the chromatin, that is uncoiled DNA, condenses into visible chromosome, each consisting of 2 sister chromatids connected at the centromere. Then nuclear envelope disintegrates, allowing spindle fibers to interact with chromosome. Then centrosomes move to the opposite poles of the cell and begin forming the spindle apparatus. Then the next phase of the M phase starts, that is called as prometaphase. During this phase, the nuclear envelope completely disintegrates, allowing spindle microtubules to interact with chromosomes. Then kinetochore protein form at the centromeres of chromosome, serving as attachment points for spindle fibers. Microtubules extend and attach to the kinetochores while others overlap at the cell center to stabilize the spindles. Then starts the third phase, that is called as metaphase. During metaphase, chromosome align at the central metaphase plate, that is also called as equatorial plane. Each chromosome's kinetochore is attached to the spindle fibers originating from opposite pole, as you can see in the diagram or picture, and this precise alignment ensures that sister chromatids will be evenly distributed to each daughter cell. Then fourth phase of M phase start, that is called as anaphase. That is basically the phase of nuclear division. During this, sister chromatids are separated as a spindle fiber attached to the kinetochore shorten, pulling chromatids towards the opposite poles. 9 kinetomicrotubules also there that elongate, pushing the poles apart and thus elongating the cell. By the end of anaphase, each pole of the cell has identical set of chromosomes, and this ensures each daughter cell receives an equal set of genetic material. Then starts the last phase of nuclear division of M phase, that is telophase, in which there is chromosome decondensed back into chromatin become less visible under a microscope. Nuclear envelope reform around the 2 sets of chromosome, creating 2 distinct nuclei. The nucleolus reappears in each nucleus, signaling the end of nuclear division. The mitotic spindle disassembles as its job is completed. And now the final phase of M phase, that is cytokinesis, starts. During cytokinesis, a cleavage furrow is formed, caused by contractile ring of actin and myosin filament. The contractile ring tightens, pinching the cell membrane inward until the cell is split into 2. And as a result, cytoplasm divides into 2, creating 2 daughter cells. So this was all about mitotic phase or mitosis process. Thank you for watching. Don't forget to like, share, subscribe and comment. Thank you.